All right, hello everybody. I'm going to go over the learning targets for the second half of the cell unit. So if you go to your learning objectives, you can see that test one is right here, and we don't want to look at test one. I see that Google Docs has got this new bookmark thing. So go ahead and click on that bookmark thing, and it takes us down to test two of unit two. So let me talk about a few things here really quickly. First of all, the cytoskeleton. There's just a few basic things you need to know about the cytoskeleton. I think it's almost the easiest part of this unit. You don't need to know a whole lot of detail. So there's three structural features of the cytoskeleton that you should be aware of: the microtubules, the microfilaments, and the intermediate filaments. The microtubules are probably the most, I'm not going to say any of one of them is the most important, but they're the ones that come up in reading and in analyses the most. The microtubules are they're tubes, as the name might imply. They're made of a protein conveniently called tubulin, and they are critical in all kinds of things. All of them, all of the cytoskeleton, are important in maintaining the cell's shape. But the microtubules are also important in cell motility, which refers to the ability of the cell to move, either internally or externally. And then they're also, the microtubules are also important in the movement of chromosomes during cell division, which we're going to learn about in an upcoming unit. So right now you don't have to learn a whole lot about that or know a whole lot about that. One thing that I do recommend knowing, though, is the idea that the cilia and the flagella. So remember that cilia and flagella, let's see, I think I have an image of them here. Cilia and flagella are ways that different cells can move externally. So the flagella is a flagellum, singular, is a tail, and the cilia are like little tiny hairs that they move quickly, and they can push the cell, they can propel the cell through whatever fluid it's in. You do also sometimes have cilia inside of a multicellular organism, perhaps to push debris out of your lungs. For example, you have cilia on your lung cells that push debris toward your throat, so you cough it out or whatever. Okay, so they, both flagella and cilia, have a similar structural design. They're made of microtubules, and they have this 9+2 arrangement. So it's 9 pairs of microtubules. So we basically, if this is a microtubule, my finger is a microtubule, and you cut my finger in half, you would see like a circular bone. Well, that's what they've done here, only they cut a flagellum in half, or a cilia, and then you're looking at the microtubules kind of coming out towards you, and so that's why they look round. And there's 9 pairs of them, and then there's a pair that are in the middle, and both flagella and cilia have that same structure. All right. And I forgot to mention, there are also the centrioles, and I think I grabbed a picture of them too, but I'm not positive. Yep, I did. The centrioles are the ones that are involved in helping the cell to divide and the chromosomes to line up in the middle of the cell, and then the centrioles help pull the chromosomes apart. The centrioles have a slightly different structure, which is interesting. Instead of that 9×2, they are 9 triplets of microtubules. So you can see as I move my arrow here that they come in triplets, and they basically they don't do much. The centrioles don't do much until the cell is ready to divide, and then they grow these microtubule fibers that are called spindle fibers that grab onto the chromosomes and let them split. You will learn more about them later. So right now, just introducing them to you, but you don't need to know a whole lot about them. Okay, and then the other thing that I want to say, I'm going to read you the little section in purple here first, and then we'll go back. It says you do not need to memorize the above terms. So this is something that might show up on an AP exam, but they're not going to ask you what do plasmodesmata, gap junctions, desmosomes, or tight junctions do, or what are their functions. They're going to tell you in the question itself. So for example, I have a sample question here. This comes directly from AP. So this is showing you how you didn't need to memorize these words, but they could still come up in a passage. So it says, in flowering plants, plasmodesmata, but now listen, it defines it for you, are narrow channels through cell walls that connect the cytoplasms of adjacent cells. An explanation of how plant cells communicate, blah, 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 blah. Okay, so they told you what plasmodesmata were. So you don't need to memorize these terms. These are just terms to become familiar with so that when they show up in passages, you're not like, what the heck is that? Plasmodesmata, that's a scary word. You've seen it before. So there are two types of junctions these are junctions that are between cells. Two of the types of junctions are called open junctions. And what that means is that cytoplasm can move in between each of the cells. So it's like a little passageway that the cytoplasm can pass through. In plants, those passageways are called plasmodesmata. In animal cells, they're called gap junctions, and it just allows the cell to transfer material from one cell to the next. The desmosomes and the tight junctions are also only in animal cells. So the plasmodesmata plants, that's it. PL for plasmodesmata, PL for plant, that's it. Those are all plants have. Animals have all three of the other ones. So tight junctions are junctions that adhere. Oh, sorry, I didn't say what desmosomes were. We'll start with desmosomes. Desmosomes are things that allow cells to be anchored to certain tissues so they don't float away, literally. So desmosomes hold cells in place. Tight junctions waterproof cells. So they make the cells so incredibly tight to one another that not even any water can get through. So that's what a tight junction is. All right, moving on, because you don't need to know a whole lot about those terms. If you're familiar with them, you're better off. Okay, the plasma membrane. We've already learned a lot about the plasma membrane, but this is a lot of reminders. So you know about phospholipids. Phospholipids, remember, we draw like a little circle, the head, and we say that that's hydrophilic. It has a phosphate group in it, and phosphate groups are charged. And so that means that it likes to be close to water. Then we draw it with two little tails coming down, like a little stick figure with two legs. And those tails are fatty acids. They can be saturated or unsaturated. Remember, if they're unsaturated, they have a little bend to them. If they're saturated, they're just straight. And the phospholipids, the lipid tails are hydrophobic, they are nonpolar, and they turn away from water. So we get this lipid bilayer. That's what the cell membrane is made of. Trying to see if there's anything more we need to know about that. And then it says, I think this is written by AP, and I think this is a little bit misleading. So it says embedded proteins can be hydrophilic with charged and polar side groups or hydrophobic with nonpolar side groups. That's a little bit misleading. If you have a protein that's completely embedded in the plasma membrane, then it has nonpolar and polar areas. Nonpolar areas are the parts that are going to be closest to the hydrophobic tails of the phospholipids, so in the middle. And then the hydrophilic heads, like opposite ends of the protein, are going to be near the heads of the phospholipids. So let me say that again, because I think I stammered on it a little bit. So if this is a protein, the top and the bottom of the protein are going to be hydrophilic because it's going to be close to water and it's going to be near the hydrophilic heads of the phospholipids. Then the middle of the protein that's embedded throughout the phospholipid bilayer, that's going to be nonpolar because it's embedded in those nonpolar tails. Now, you could theoretically have a protein that's completely embedded in the cell membrane, and that one would be hydrophobic, or you could have some proteins that are entirely hydrophilic, and they would be sitting on either side of the plasma membrane. So they would sit either at the top or at the bottom of the plasma membrane, but they wouldn't go all the way through. So I felt like that was a little bit misleading. Okay, describe the fluid mosaic model of cell membranes. So fluid mosaic. Fluid means that it's constantly moving and shifting. The way they say the fluid mosaic model, they compare it to icebergs in the water. That icebergs are shifting and moving with the currents and they shift around. It's not a stationary or static structure in the cell. Mosaic just means that it's this mosaic of all these proteins and other things that are embedded in the cell membrane. So it's constantly moving. I often tell my students, if we take a picture of the cell membrane and then 1 second later we take a picture again, all the proteins will have shifted because they're constantly moving around. All right, see if there's anything we need to know. Oh yeah, don't forget about the steroids. So we learned specifically about cholesterol. So to back up, remember our biochemistry unit, a steroid is a type of lipid trying to get you to remember all this because you need to remember all of this. So lipids, remember, are nonpolar molecules that are primarily carbons and hydrogens. In the case of a steroid, it's a bunch of fused rings, as opposed to the carbohydrates, which had rings. I'm trying to make rings with my fingers here. They had rings, but they were joined with like a glycosidic linkage. So there was like a ring, and then there was this part that stuck down. The steroids are like this, like the rings are totally fused together. So carbohydrate is a type of steroid. It is embedded in the phospholipid bilayer, and it allows cell membranes to retain the right amount of fluidity depending on the temperature. So they prevent the lipid bilayer from getting too fluid in very warm temperatures, and they prevent the lipid bilayer from becoming not fluid enough in cold temperatures. So they moderate the fluidity of the lipid bilayer. So cholesterols within the lipid bilayer are good. Then there were the glycoproteins and the glycolipids. So glyco is a reference to like carbohydrates. So when you look, let's look at a picture of the plasma membrane right here. Oh, did I bring it? I didn't open a big picture of that, or did I? I don't know if I opened a big picture. So let's open this in a new tab. And I didn't. So there we go. So just to remind you really quickly, let's see if I've got a good picture of a glycoprotein. Here's a glycolipid, a good picture of a glycolipid on this side. So it's labeled glycolipid. Notice that these are the rings of a carbohydrate. So it's got a whole bunch of rings of a carbohydrate, but they're attached to the two lipid tails of the phospholipid bilayer. So that's why it's called a glycolipid. Pretty straightforward. And then a glycoprotein, it's labeled, here, I'm sorry. I was thinking it was this green thing here, the blue thing. This is the glycoprotein. So here are the rings of the carbohydrate, and then it's attached to one of the embedded proteins, one of the integral proteins in that case. Hey, this is also a good image for me to show you that hydrophilic, hydrophobic thing I was talking about. In this embedded protein right here, this part of the protein here is going to be hydrophobic, at least on the outside. This part here is going to be hydrophilic, and that's why it sits the way it does in the cell. One thing we may or may not talk about in just a minute is that if this is a channel protein, then the hallway that goes through the middle, the hole that goes all the way through the middle, that's going to be hydrophilic as well. So it'll allow charged or polar molecules through it. But the area that's closest to these hydrophobic tails, that part of the protein is going to be hydrophobic also. All right, whoops, Moving on, let's see what else we need to talk about. Membrane permeability. So we saw in the lab that we've been doing, the dialysis tube, remember that in the beaker, and we put sugar starch solution in there. We called it semi-permeable. Semi means it's a little bit permeable, and that just means that small things can pass through and big things can't. Selectively permeable is a similar concept, but it implies intelligence, that it's selecting what comes in and out. Okay, it's not really intelligence, but the cell has the ability to determine what it's going to let in and what it's not going to let in. So that's why we call it selectively permeable. So whereas the dialysis tube, it's just little holes and that's it, and it has no choice what comes or goes, just little things come and go. With a cell, if certain things can't pass through on its own, then it has embedded proteins that can allow it to pass through or can even push it through. So the cell is selectively permeable. It can choose what to bring in or bring out. All right, cell membranes separate the internal environment from the external environment. Isn't that a duh? Why does AP need to write that? I guess it's important, but selective permeability is a direct consequence of membrane structure as described by the fluid mosaic model. Here we go. This section is about that worksheet that you did in small groups, and then I helped you kind of finish up. I think it's called membrane functions, I think was the name of the little packet. So small nonpolar molecules, nitrogen, oxygen, carbon dioxide, can pass freely right through the membrane. So if I were going to pull out that image again, Carbon dioxide, oxygen, and nitrogen can pass right in between these phospholipids. They go straight through the cell membrane. They don't need any kind of help whatsoever, and they go through efficiently. Hydrophilic substances, such as large polar molecules, sugar and bigger, and ions, so things that are fully charged, move across the membrane through embedded channel or transport proteins. So they can't pass through the phospholipids. They have to go through these integral channel proteins. And then we learned about this: small polar uncharged molecules, like water, can pass through the cell membrane, but in very small amounts. So water can go in between the phospholipid bilayers. They can pass in between, but they don't do it very efficiently. So I'm just reminding you of this. There are proteins called aquaporins that water can just zoom right through. It's polar on the inside, and the water can travel right through those aquaporins. Have I mentioned I like those words? Okay, moving on. The cell wall, in the importance of the cell wall, maintaining cell structure and function. So in a plant cell, the cell wall, plants don't have skeletons. So the cell wall is what allows plants to stand upright. And we're talking about non-woody plants. Wood is a whole other thing that we could talk about another day, but green plants. And turgor pressure, remember, is super important to those cell walls. So the central vacuole has to have a lot of water in it pushing against the cell walls. And as long as there's a lot of turgor pressure, aka water pressure, inside the cell walls, then the plant can stand upright. So it's important in maintaining cell structure and function. Permeability so they're similar to cell membranes. And then the cell walls of plants, prokaryotes, and fungi are made of complex carbohydrates. And just so you have the words kind of in your playbook, is that a word? I don't know. Cell walls, remember, of plants are made of cellulose. Prokaryotes, they have a complex carbohydrate called peptidoglycan. Just don't be freaked out by the word. You've heard it. You don't need to memorize it, but that's the word. And then fungi, like mushrooms, they have a cell wall, but it's made of a carbohydrate that's called chitin. It's spelled Chitin. All right, membrane transport. Describe the mechanisms that organisms use to maintain solute and water balance. So the first thing that we talked about was passive transport. If something is passive, it means that it did not use any kind of ATP. It didn't use any kind of energy. When you're feeling passive, you're usually not expending a whole lot of energy. If you're being active, then you're using a lot of ATP. Okay, so passive transport is usually the diffusion of molecules from an area of high concentration to an area of low concentration. So imagine a sled going down a hill. When the sled goes down that hill, you don't have to add a whole lot of energy. It just goes down the hill. If you want to go from the low end of the hill to the high end of the hill, then you have to push that sled up, and that takes energy. So that's going up the opposite direction, and that requires ATP. So let's get back. Sorry, I'm going ahead of myself. So passive transport is going from high to low. So keep clear what I mean by high, a higher concentration. So if we were to use a percent, for example, if I were to say the water on one side of the cell is 90% water, and the water on the other side of the cell, on the inside of the cell, is 80% water, then the high is 90% and the low is 80%. And so the water is going to diffuse or osmose from 90% to the 80%. Okay, and no need to add any kind of ATP or direct metabolic energy. Passive transport plays a primary role in the import of materials. So, for example, bringing in oxygen and bringing in water, and the export of wastes just happens passively. I want to remind you too, it comes up in a minute, but I don't know why it's not written right there. Facilitated diffusion is also diffusion going from high to low. It also doesn't require energy, but it does require the use of one of these membranes as a channel. So if this is a channel protein, and it's basically just a hole going through the protein and molecules can pass through, no ATP is necessary, then that's a type of passive transport. It's just listed separately as though it's not part of passive transport, but okay. Passive transport, blah, blah, blah. Active transport requires the direct input of energy to molecules to move from low to high. So pushing that sled up the hill requires lots of energy. If you want a lot of hydrogen ions on one side of the cell membrane, they're going to have a tendency to come in. They're going to have a tendency, let's say, that didn't make sense. Let's say on the inside, you don't want many hydrogen ions, and on the outside, you do want lots of hydrogen ions. They're going to have a tendency to diffuse in or to come in through protein channels. But we want to push them out. So we want them to go from low concentration to high concentration, and that takes ATP. We call that active transport. If the cell has to use ATP to get it out, then that's called active transport. All right, and then transporting large molecules, it's also called bulk transport. Bulk means big. So across the plasma membrane, and there we're talking about endocytosis and exocytosis. And I have this cool video that I can't remember. I don't think I showed you guys. And so when I say cool, let's be realistic. It's not that cool. This is like the driest video I've ever seen in my life. I'm skipping right to the part that I think is important. There's no cool music, and the narrator is like blah 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 blah, like as boring as you could possibly be. But I want you to look at what they're showing here, because I think it's so like you start to get the hang of something. This is a Golgi that we're looking at right now. And so just listen, watch what it's going to package something into a vesicle. And then they're going to look at it. They're going to turn that vesicle. They're going to look at it at the molecular level. And you'll start to see the phospholipid bilayer. And you'll be like, oh, now I get it.
Packaged in a vesicle, which moves to the plasma membrane.
So
as membranes merge again, the contents leave the cell without actually crossing the plasma membrane.
And that's exocytosis. Pretty cool, right? It just merged with the cell membrane, and now endocytosis is just the opposite.
This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Excellent. So very boring video, but I think it's the kind that makes you go, oh, now I get what's going on there and how that all works. All right. Internal vesicle fuse with the plasma membrane, secrete. There's that word secrete. When we saw it going out, it is secreting it. You also can get rid of waste that way. Endocytosis, bringing stuff in, blah, blah, blah. Okay, facilitated diffusion, I already mentioned a little bit. Explain how the structure of a molecule affects its ability to pass through the plasma membrane. So remember that small nonpolar can get through. Large polar cannot, so they have to be helped through. Let's see if there's anything charged and large polar molecules through the membrane. Large quantities of water pass through aquaporins, so we know those, because that's my favorite word. Charged ions, including sodium and potassium, we're going to watch a video about that in just a second, require channel proteins to move through the membrane. And then membranes may become polarized by movement of the ions across the membrane. We'll talk about that a little bit more later. But when you have lots of positives on one side and lots of negatives on the other side, you actually polarize the whole tissue, which is kind of cool. And then membrane proteins are necessary for active transport. We know that. I'm not sure how that's facilitated. How is that facilitated diffusion when it's talking about active transport? They're not the same thing. Moving on. Metabolic energy, such as ATP, is required for active transport of molecules and or ions across the membrane and to establish and maintain concentration gradients. The sodium-potassium, what the heck is this, KJ? The ASE ending is the ending for an enzyme, which is a type of protein. So this literally means the sodium-potassium pump, but it's another word for it. So contributes to the maintenance of the membrane potential. I just want to remind you really quickly, we saw this already. I fast forwarded it just to 1 minute. You can watch how the sodium-potassium pump works and why this is called active transport. Notice the use of ATP.
Free sodium ions.
Oops, I still have it really, really fast, so maybe I'll slow it down to the correct speed. I'll slow it down just a smidge. Let's go back to just a teeny bit right there. We'll try it.
After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate.
It lost a phosphate.
This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, two potassium ions positioning themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released.
I'm going to say a bunch of things there just to make it feel really clear. First of all, remember that a channel protein is just a hallway. A carrier protein is one of these things that changes shape. And you know this from our biochemistry unit, why a protein might change shape. It has to do with the charges that are being added near the different functional groups. So as it adds ions, it's causing the folding, the tertiary structure of this protein to change depending on which ions are in what positions on the protein. And so the protein is changing shape because of that. So when it's bringing ions across to either side, that's called a carrier protein, as opposed to a channel protein, which doesn't change shape. It's just basically a hallway that molecules can pass through. And aquaporin is an example of a channel protein that allows water to pass through. All right, tonicity and osmoregulation. Oh my gosh, big fancy words. Okay, so explain how concentration gradients affect the movement of molecules across membranes. So I'm just going to give a quick aside that your textbook doesn't talk about water potential right now, which I think is unfortunate because AP puts water potential right here in this particular unit. So if you look at page 596 to 598 in your textbook, you're going to see a discussion of water potential. I'm going to have you, if you haven't already, watch a separate video from Bozeman Science where he talks about osmoregulation and talks about water potential. And then we'll do a couple worksheets just to make sure you feel the math is actually very easy, but just to give you confidence on it. So we'll do those a little bit later. So for the 2021 school year, the distance learning school year, we are not going to have water potential on this test. I'm going to give you a quiz about it later, but it's not going to be on this test. Future years, if you're watching this video, it will be on the test. All right, so I'm up here again. External environments can be hypotonic, hypertonic, or isotonic to internal environments of the cell. So let's practice those words. Gosh, I should have typed that in. So let's try hypotonic solution. I'm hoping we'll have a comparison. Oh, we found a comparison one. This is perfect. Open it in a new image, in a new tab, and let's take a look at these. Okay, so these are awesome. This is exactly the image that I wanted. So in a hypotonic solution, remember that I had that student say, well, I think of hippos because hippos are these big bloated African creatures. So in a hypotonic solution, there's more water on the outside than there is on the inside, and water rushes in. The perfect example of a hypotonic solution, because it is always hypotonic, is distilled water. Distilled water is 100% water. It cannot be anything other than a hypotonic solution. Perhaps it could be an isotonic solution, but it could never be a hypertonic solution because its concentration is the highest. It's the highest hill you could possibly have at 100%. So nothing's ever going to go higher than 100%. So water rushes into these cells. Now, in an animal cell, that's a problem because we have these soft, squishy cell membranes. So if water rushes in, just like with a water balloon, if you fill it too much with water, it pops. So a hypotonic solution, bad for an animal cell. Interestingly, however, it's great for a plant cell. Plant cells, remember, have this cell wall, and they want high water pressure. They want high turgor pressure inside. So a hypotonic solution that increases the water pressure or the turgor pressure inside, good for the plant. Isotonic solution for an animal cell, that's ideal. Iso means same, and it basically means that the concentration of water and dissolved substances inside and outside are the same. So water or dissolved substances, but primarily water, is going to be coming and going at an equal rate. We call that equilibrium, and the cell won't change size. A hypertonic solution is bad for both types of cells, bad for both plant and animal cells. A hypertonic solution is when there is less water on the outside than on the inside. The percentage of water is less on the outside than it is on the inside. So the example, this is without we're going to talk about water potential very specifically, but just without talking about the actual numbers. Let's give you some percentages. So let's say this cell was 80% water on the outside, but let's say on the inside, 80% water and 20% dissolved substances. And let's say that we put it in ocean water, and let's say ocean water, I don't know what number it is. We'll say 75% water and 25% salt. It's not that low, but whatever. And make it a point. So 85 on the inside, 75 on the outside. It's going to go from high 80 to low 75. So the water is going to leave the cell, and the human cell, the animal cell, is going to shrivel up. They can't work efficiently that way. This is why you don't drink ocean water when that's the only thing available. Does it matter if you're swimming in the ocean and you drink a little ocean water? No, not at all. What matters is if you have no other water available to you and you try to subsist on ocean water, it will actually pull water out of your cells and make you more dehydrated. Crazy, right? Drinking the ocean water makes you more dehydrated than if you just didn't drink anything. And then this is also why you get thirsty when you're eating salty foods like potato chips or French fries or whatever. And then same idea for the plant cell. This is what is a huge building environmental disaster, literally, in Minnesota. We add salt to our roads every year and to our sidewalks. That salt never goes away, and it's getting washed into rivers and lakes and into our gardens. And it basically creates ocean water when the snow melts and the rain comes and whatever. And so that pulls the water out of plant cells. It would also do the same. It would also desiccate animal cells, so fish and snails and other animals that live in the water as well. So as our salinity goes up in our freshwater lakes, that is bad news for our plants and animals that depend on those environments. All right, we're getting near the end here, you guys. Osmoregulatory, just to be clear, osmo refers to water, osmosis, and then regulation refers to controlling how water comes and goes. So let me give you an example. There's that single-celled organism. I think I just saw a picture of it somewhere. The single-celled organism called paramecium. Give me a second. Paramecium. And suddenly I forgot what her thing is called. We'll just go with paramecium and see if it says it here. Vacuole, there we go, the contractile vacuole. So remember that the paramecium, I was trying to find a good image of it, the paramecium has this thing called the contractile vacuole that allows her, I always call them hers for some reason, that allows them to push water out. So that's osmoregulation. If water is rushing in, the paramecium is able to push water out. So it's able to regulate how much water is in. Aquaporins, do that also. Okay, growth and homeostasis. You definitely need to become familiar with the word homeostasis. Same. So just maintaining a constant internal environment, that's how cells do best. So keeping your body at a normal temperature, good. Keeping your water levels at a normal level, good. Keeping your sugar levels at a normal level, good. Anything that throws our body out of homeostasis, bad. All right. So then there's these are the equations, and we'll talk about those another time. And then we're almost done, you guys. And I think this is dumb. Like, why is this here at the very end? Mechanisms of transport. Describe the processes that allow ions and other molecules to move across membranes. A variety of processes allow for the movement of ions and other molecules across membranes, including passive and active transport, endocytosis. Didn't we just say all that? Why is it here again? I have no idea, but there you go. So you guys, we are all done. Let me know if you have any questions.